Good morning from Australia. Welcome everyone to the 15th session of the UNESCO GO2NE, or the Global Ocean Oxygen Network webinar series. My name is Jodie Rummer. I'm a professor of marine biology here at James Cook University in Australia. Here where I'm based in Townsville, we honor the Wilguru Kaba and the Bindal peoples, the traditional custodians of the land from where I'm speaking today and from where I work. And we honor their elders, past, present, and emerging. Indeed, a very good morning from Australia, but good afternoon or good evening from wherever you're calling in from. The whole GO2NE team is very happy that you took the time to be with us and our incredible speakers, whether it's morning, afternoon, or evening for you. Before we start, a couple of announcements. Firstly, if you haven't already seen it, head over to the GO2NE website, it's a really good website, to download the first issue of Good News, which is our quarterly newsletter for the ocean deoxygenation community. And secondly, the next GO2NE webinar has not yet been scheduled, but please keep an eye out for details to register when it will be announced. We're really excited about that. And a bit of housekeeping. You as the attendee have the possibility to type in your questions in the question box. And the chat can be used to share additional information, or if you have any logistical problems, and I'm going to keep an eye on it throughout the whole session. So please use that chat box. We're really excited for questions and information. So without further ado, we have two really great scientists speaking with us today. We are going to start with Ariel Pesner, who is based at the Scripps Institute of Oceanography with the University of California at San Diego in the US, and then followed by David Suggett, who is at the University of Technology, Sydney here in Australia. Both speakers will present for about 20 minutes, followed by a 10-minute question and answer period. So first, I will introduce Ariel Pesner, and Ariel, I'll ask you to start sharing your screen. So I first learned of Ariel's work just over the past few months at one of our GO2NE workshops. And I was just really, really impressed by how dynamically she spoke about issues that, of course, coral reefs, our theme for today, are experiencing with low oxygen issues. And as an early career researcher, and I'm told that the PhD defense is only a few weeks away, I was super excited for her to present her research to us today. So without further ado, Ariel, I will ask you to take the screen. Thank you.
Perfect. Thank you, Jodie, so much for the introduction. And thank you everyone for having me here today and for organizing this seminar. I'm really excited to share some of my work with all of you. As Jodie mentioned, this all is part of my PhD thesis work. So I'm sharing just one chapter with you here today. And that was looking at coral reef hypoxia, present-day exposure, and projections under forming. So traditionally, coral reefs are assumed to be well-oxygenated systems. They have these crystal clear waters and a high abundance of primary producers, which we would expect to mean that there's lots of oxygen available in the environment. However, recent evidence from hypoxic events at several coral reefs around the world have shown that hypoxia can occur on tropical coral reefs, and it can lead to mass migrations of reef invertebrates towards the surface, as well as bleaching and mortality of the corals themselves. And hypoxia on reef can be caused by a variety of different factors, which Nelson and Altieri grouped into two kind of big categories, one being factors that increase oxygen demand, such as temperature rise, which increases metabolic rates, and thus biological oxygen demand, as well as factors that lead to eutrophication, such as coral spawning slicks or nutrient pollution or algal blooms, which also draw down oxygen. And there can also be factors that prevent re-oxygenation. So this could be the geomorphology or the shape of the reef, how shallow it is, if it's prone to stratification, if there are different weather conditions that lead to little to no mixing, like slow current speeds, or if the reef is somewhere with extreme tides that kind of isolate the reef from the rest of the open ocean. And so on a global scale, we know that coral reefs are experiencing declines in coral cover. Over the past few decades, of course, we've seen this decline in hard coral cover across the globe. And a lot of this can be attributed to changes in temperature, which have led to mass coral bleaching events, which can lead to coral mortality, especially during El Nino years. However, of course, corals are facing a lot of different environmental drivers that may be stressing them, such as nutrient rise, which is leading to overgrowth of algae on coral reefs, increasing CO2 concentrations from release of atmospheric CO2, which is leading to ocean acidification, which may have consequences for marine calcifiers such as corals. However, the role of oxygen in driving some of these changes in coral reefs has not been as well investigated. And so in the open ocean, trends of deoxygenation are relatively well constrained. From model estimates, we know that in both the surface and the deep ocean, there's been this kind of general trend of oxygen loss across the global oceans in the past few decades. And a lot of this has been attributed to changes in stratification due to warming, as well as decreases in oxygen solubility in warmer seawater and changes in respiration rates. However, it's likely that we're actually underestimating the occurrence of hypoxia on tropical reefs. This figure from Altieri et al. 2017 is showing the overlap between areas that we know are documented dead zones as the red circles and where we can find coral reefs around the globe in purple. And despite the significant overlap in a lot of these regions, only the areas in yellow have been places where we know that hypoxia has led to coral reef mortality. And since there are so few of them and the overlap with the dead zones is so high, it's likely that we're actually missing a lot of these hypoxic events. And so the authors posit that there's several reasons for why this may be. One being the lack of long-term monitoring of dissolved oxygen on reefs, or maybe we're just not reporting it very often. Traditionally, hypoxia research has been mainly performed by researchers in temperate regions, and so the research is being done in temperate systems. And then also in terms of coral reef researchers, we tend to focus on more healthy reefs or well-studied reefs instead of maybe the reefs that are more likely to experience hypoxia. And also in this study, they did an analysis to kind of predict how many reefs around the world would be at elevated risk to hypoxia based on their geomorphology or exposure to different anthropogenic pressures. And they found that about 13% of global reefs are at elevated risk of hypoxia. But we didn't really have any of the data to confirm this until this study. So the goal of my work was to compile a global coral reef oxygen data set to kind of address some of the gaps in the knowledge that we have about hypoxia in coral reefs. And so this data set included 33 reef sites at 12 different locations around the world. These 12 locations are indicated here in the center of the map, and then each of the 12 locations are plotted with these satellite images to show you that we had multiple deployments within some of these locations, which led to our 33 reef sites. And importantly, these reef sites covered a variety of different coral habitats and different types of coral reefs to kind of extend our analysis to not just one type of reef. So we had fringing reefs, we had kind of patch reefs, we had reefs that were adjacent to seagrass beds, as well as deeper core reefs. And also the majority of these data sets were recorded by CFOX sensors, which use Andera oxygen optodes to record high frequency oxygen concentration data. And I, of course, want to highlight that this was an extremely collaborative effort. And the data that's presented here was collected by a ton of different researchers at a variety of different institutions around the world and supported by a variety of different funding sources. And of course, this work wouldn't have been possible without all of their help. And so the motivating questions now that I had this data set were to first look at how does baseline oxygen variability differ between reef sites around the world? So what do typical oxygen conditions look like on these reefs and how do they compare? And then using this data, looking at how frequently do these global reefs experience what we would consider hypoxic conditions under present day conditions, and then also looking at how hypoxic event intensity, duration, and severity would change in a warming world. So before I get into that, I wanted to focus a little bit on what we determine as hypoxia thresholds. So we know that the effects of hypoxia can range from sublethal to lethal. So on the sublethal side, we can observe changes in respiration, photosynthesis, or calcification in the corals, and in the other reef invertebrates, changes in behavior. And then, of course, on the lethal side, we would observe changes in mortality. And unfortunately, we don't have a lot of data for this for tropical reef organisms. And so there is a lot of data from the temperate literature. And from the temperate literature, we know that the thresholds of hypoxia actually vary widely between taxonomic groups. And so looking on the left here at the median lethal concentration, we see that, for example, gastropods have much lower lethal concentrations than crustaceans. And when we look at sublethal thresholds, we see an even greater spread, both within and between taxonomic groups. And traditionally 2 milligrams per liter, or about 61 micromoles per kilogram, has been defined as this kind of universal static cutoff of what is defined as severe hypoxia or not in seawater. However, we argue that this static threshold maybe doesn't encompass all of the relevant thresholds for all of the relevant species, perhaps on a coral reef especially. And it also doesn't capture a lot of the sublethal thresholds, which may still have an impact on the community composition and community functioning if we were to observe just sublethal effects of hypoxia. And so for the purposes of the study, we introduced 4 different thresholds to kind of assess how our assessment of hypoxia would change if we had different thresholds available. And so on the upper end, we had a weak hypoxia threshold, which was at about 5 milligrams per liter or 153 micromoles per kilogram based on this study. And then we introduced 2 other ones that were equally spaced. So we have severe, moderate, mild, and weak hypoxia thresholds. And so looking at the oxygen data for our 33 different reef sites, we first wanted to look at just what the oxygen availability and variability looked like on these reefs. And so this figure on the right is showing our 12 different locations with the multiple sites plotted within each location. And then the left panel is showing dissolved oxygen concentration, and the right panel showing dissolved oxygen saturation. And of course, we have our 4 different thresholds as these dotted gray lines. And we can see that the mean and extreme dissolved oxygen conditions vary widely between these reef habitats and even between sites at the same location. So if we look, for example, at Dongsha Atoll, the oxygen conditions experienced at these reefs here are extremely variable. They have both very low oxygen concentrations and very high oxygen concentrations during our deployment time. Whereas if we look at a place like Tutuila, for example, which is a bit of a deeper fore reef, we see that the oxygen concentration here is much more modulated and hovers around a singular value for the entire deployment. And we can also look at this in terms of time. So this is a climatology plot looking at the mean dissolved oxygen value for each of our sites over different hours of the day. And we see this nice diel trend where we have low oxygen in the early morning and higher oxygen in the late afternoon, which we would expect as signals of respiration and photosynthesis during the daytime. However, you can see that the amplitude of these changes are very different for different reefs around the world. For example, Dongsha Atoll has this very large amplitude where the reefs here are experiencing what we would call severe hypoxia almost every hour or every day of the deployment. Whereas a reef like Bocas del Toro, for example, at least while we were measuring it, was experiencing much smaller amplitudes in oxygen from day to night. And so this establishes that these reefs are having very different environmental histories of oxygen. And so perhaps these static thresholds may not be as applicable when we compare between different reefs that already have very different oxygen variability under present day conditions. And so next we wanted to ask what might happen to reef oxygen under warming. And so to illustrate this, we can think of kind of a typical coral reef that has reef invertebrates, it has corals, and may have some primary producers. And of course, we would expect to see this diel cycle, as I mentioned, where we have high oxygen in the late afternoon as a result of photosynthesis and low oxygen in the early morning, perhaps as a result of respiration. And perhaps this reef experiences some level of hypoxia just a little bit in the early morning hours. However, if this reef were to warm, we would expect not only for the oxygen solubility to decrease just physically in the seawater, in warmer seawater, but also an increase in metabolic rates and thus respiration rates, which would draw down additional oxygen. And these two would both act to draw down the available oxygen that's available on these reefs. And so we were predicting that the overall trend of oxygen on these reefs would decline under warmer scenarios, and thus this reef would maybe experience more hypoxia more of the time under warming. And so to test this and quantify this in our data set, we leveraged location-specific temperaturized data from the CMIP6 projections, which use these shared socioeconomic pathways, or SSPs, which are these 4 different warming scenarios based on different mitigation efforts for climate change by the end of the century. And so from this, we extracted mean end of the century warming predictions for each of our 12 different locations for these 4 different scenarios. And so in addition to these 4 different temperature projections, we also added a more acute, intense scenario of a 6-degree heat wave as our 5th scenario. And then we calculated the decrease in oxygen solubility on these reefs under these warming conditions, as well as the increase in reef respiration using a Q10 relationship to then predict what oxygen would look like on these reefs under warming. And so from this data, we found that warming will overall reduce the available oxygen on reefs, leading to increased exposure to hypoxic conditions. So this figure is showing on the x-axis all of our 33 different reef sites, and the y-axis is dissolved oxygen concentration. So each of the dark shaded bars represent the mean range of oxygen experienced on these reefs in blue in present day to our increasingly warm scenarios up into red in the severe heat wave scenario. And then the lighter shaded bars represent the extreme ranges. And so if we look at one site at a time, for example, looking at Hog Reef, which is in Bermuda, which is a more temperate reef, we see that the oxygen concentrations at present day are relatively high. And also the variability experienced here is a little bit smaller than some other locations. And even under the most severe heat wave scenario, we see that while the oxygen concentration does go down on this reef, it's still well above our hypoxic thresholds. But if we look again at Dongsha Atoll, for example, we see that this reef, which is adjacent to a seagrass bed, that under present day conditions, this reef already experiences every level of hypoxia based on our set thresholds, and that under this most severe warming scenario, this reef would actually be hypoxic, at least weakly hypoxic all of the time. And so overall, we found that 84% of the reefs that we surveyed here would experience weak to moderate hypoxia or already experience weak to moderate hypoxia, and that about 13% of these reefs experienced some duration of severe hypoxia under present day conditions, which matches pretty well with the projections made by Altieri et al. In 2017. And then looking at end of the century scenarios under warming, if we look at the most severe horrible climate change scenario, the SSP5, we see that almost all of these reefs would experience some level of hypoxia, and about a third would experience severe hypoxia by the end of the century. We also found that warming would increase the duration of hypoxic events across many of our reefs. So this figure is showing the percent of sites that experience hypoxia of varying durations from less than 1 hour to greater than 24 hours for our 4 different hypoxia thresholds. And generally, we see that regardless of which threshold you look at, we see this shift where under warmer conditions, a greater percent of these sites would experience longer duration hypoxic events. And so this is important because not only is it the concentration of oxygen that these organisms are exposed to that may determine how they are impacted by hypoxia, but it's also the amount of time that they're exposed. And from the temperate literature, we know that the median lethal time of exposure between different taxonomic groups can vary by orders of magnitude. And so not only is it important to look at the concentration, but perhaps also the duration of time that these organisms are being exposed to understand how this may affect coral reefs ecologically. And so altogether, we found that warming would increase the total duration, intensity, and severity of hypoxic events across all thresholds for all of our reefs that we surveyed here. And so regardless of the threshold that you look at, we see an increase in duration, an increase in intensity, which is the difference between the threshold that we set and the oxygen that you would observe, as well as severity, which is an indicator which combines both duration and intensity. And so we would see this increase under these different warming scenarios for all of our sites. And so overall, we found that present day means and extremes of oxygen vary widely, both among and within our reef locations, and highlight how important environmental history may be in driving how sensitive these reefs are to changes in oxygen. And we also found that hypoxic conditions are already pervasive across reefs around the world if we use the thresholds that we have already set, and that under warming, we would see an increase in the duration, intensity, and severity of hypoxic events by the end of the century. So what does this really mean for coral reefs, right? That's the bigger question. Well, from the little data that we have from tropical coral reef hypoxic events, we know that hypoxia can lead to whole community shifts. So this is data from Maggie Johnson and colleagues looking at a hypoxic event in Bocas del Toro, and they found changes in the left here. This is changes in the coral community composition, both before, during, and after a hypoxic event, as well as on the right, changes in the entire reef community before, during, and after. And while we see some level of recovery, perhaps in some of these groups before, during, and after, we know that we can see changes in the entire community that maybe aren't even resolved 1 year after exposure. And so if these reefs are experiencing, maybe more than one hypoxic event per year, what would these changes look like? It's something that really needs to be studied more. And unfortunately, to make these predictions, we also need to know what relevant thresholds are for organisms, especially in tropical coral reefs. We have a lot of data for temperate species, but this figure is showing that we really don't have a ton of data for tropical coral reef species. And of the data that we do, we already see that there's a large range in the relevant thresholds, especially looking at sublethal or lethal thresholds between different taxonomic groups and even within them. And so understanding the differences between lethal and sublethal thresholds, as well as maybe incorporating some aspect of time like duration or frequency into these studies, I think would be really important for allowing us to be able to predict how oxygen and hypoxia will affect reefs kind of as a whole community moving into the future. And I also wanted to highlight too that warming and hypoxia are not occurring in isolation. So a lot of the factors that lead to hypoxic events in coral reefs, like these slow flow conditions and warm conditions that lead to more respiration, also lead to acidification, as respiration releases a lot of extra CO2. And so meta-analyses have shown that hypoxia and acidification, when they co-occur, their effects can be additive. And further, under warmer conditions, organisms, at least in temperate studies, have been shown to increase their sensitivity to low oxygen. So we need to incorporate kind of these multi-stressor contexts into some of our studies as well. And so altogether, we believe that this may have severe consequences for reef and reef-associated taxa, perhaps depending on which reef you are and what their environmental history looks like. And so we make recommendations in the paper that's currently in review, looking at that we need to continue long-term monitoring of reef-dissolved oxygen and report it perhaps to a common database like the GO2NE database that was proposed recently. And we need to determine relevant oxygen thresholds for these reef organisms, both with the lethal and sublethal thresholds, and also consider duration, intensity, and frequency of these events, as well as maybe multi-stressor contexts. And I think overall, it's really important to understand the role of environmental history in determining these oxygen thresholds. As we know, the baseline oxygen conditions are very different between different reefs, and so that may affect how we quantify this moving forward. And so I just wanted to highlight that this ties on to a little bit of my other ongoing work using seagrasses as natural laboratories. Some of this work, again, at Dongsha Atoll, which seems like a pretty unique place. That's part of my thesis work. I'm looking at the effects of this highly variable and oxygen pH environment on coral growth. And also I've been working on other projects looking at the physiological and genomic effects of hypoxia exposure on corals. So yeah, I'm happy to take any questions, and thank you for listening.
Wow, Ariel, that was such a fantastic talk. And yeah, definitely ready for that PhD defense. It was really, really well communicated. Thank you so much for that. We have a lot of questions for you, which is super exciting. One of the questions that popped up almost initially, which I think is a great topic for us to be discussing as a community over the next little while, not necessarily for you to have the answer to, is from Gilles, who asks, do we have any working groups working on coral reefs in West Africa? And so, something that we have chatted about as a whole community is getting some of these working groups together that focus on different geographical areas. And I think that one thing that you really highlighted so nicely in your talk was that different geographical areas are experiencing, different variability in the oxygen levels and duration that they're experiencing. And so I think that that's definitely a topic that we all need to be thinking about, different working groups around the world. So not necessarily one for you to answer, but I just wanted to throw that out there. We do have a couple questions from Nicholas. And Nicholas asks, are these variations of oxygen during the day do they have an explanation? Does that suggest breathing from some organisms?
Yeah, that's a great question. So, on any reef around the world, if we put out an oxygen sensor, we would observe these diel patterns in oxygen. And that's just because, corals themselves, right, have these photosynthetic algae in their bodies. And also there's also lots of other primary producers on reefs like algae or seagrasses or whatever. And so during the day, we have photosynthesis happening when there's sunlight available. And so these oxygen values would go up. And then at nighttime, all of these organisms are respiring and taking up oxygen. And so we would expect the oxygen to go down. So these diel cycles are pretty well established on reefs. It seems like the magnitude to which the oxygen is changing from day to night really varies between these different reefs around the world.
Definitely. Nicholas's follow-up question was about biomass. So, beyond the actual coral and monitoring on that. So maybe, Nicholas, you can touch base with Ariel through some of her social media channels at some point to chat with her more about that. Emma Pontes has some comments, really nice talk. Ariel, did you happen to look at community composition for your reef sites? I'm curious if reefs with higher oxygen variability have different coral or invertebrate species than sites with lower oxygen variability?
It's a great question. It's definitely something we've thought about and we're trying to incorporate into these analyses. But unfortunately, like we just don't have community composition data for some of these sites. But it's definitely something I'm working on doing because I would agree with you that I think to some extent depth and also community composition and a lot of other physical factors like current speed and residence time and stuff also all have an effect on what these oxygen patterns look like on these different reefs.
Absolutely. It's so connected, hey? Our next question, yeah, thanks for that great question. Our next question is from Domingos. The increase in temperature of the global ocean causes degassing, namely the release of carbon dioxide and oxygen into the atmosphere. The released carbon dioxide, being denser, forms a carbon dioxide-rich layer near the surface. Could this be one of the causes of the formation of heat waves that impact the continents?
Wow, that's a great question. I'm not an atmospheric chemist, so unfortunately I'm not confident in my answer to that. I mean, I don't know. I think you'd have to consult someone who's more on the air-sea gas exchange side of the research, but it's definitely an interesting question.
I'm also not an atmospheric chemist, so I'm with you. Throw that out there to the rest of the community. Maybe someone has an answer for Domingos. Veronica asks if you can talk a little bit about the process of compiling this massive data set. I mean, it looks like so much work, and obviously it is. How did you identify the different research groups, and what was the general response when you identified relevant data but didn't obtain access? So I think more of a compiling data approach.
Definitely. It was a huge effort. It was my quarantine project of 2020, but luckily the lab that I'm in, Andreas Anderson's lab, we have a lot of this data kind of sitting around. And I think this is probably true for a lot of reef scientists who own sea foxes or any other CTDs that, measure oxygen. And so we had a lot of this data that just really had never been published or used for anything. And so a lot of this data came from our lab. And then we have, many collaborators that we worked with over the years who also had oxygen data that they can contribute. And, I tried to look through the literature for some data as well, but it was hard to, kind of take all the data that was available when especially I wanted to make sure it was all collected in the same way, using similar sensors or, for similar durations of time or things like that. And also, of course, quality control on, many different data sets is a lot, and we don't all, calibrate our oxygen sensors the same way. And so that's also a recommendation we make in the paper is that, standardizing how we collect and calibrate and report and QA and QC this type of oxygen data is also really important. It was a lot of collaborative work. All of the people I listed are co-authors on this paper. And I'm sure there's many data sets that I've missed that are probably out there, but that's why we need some sort of like centralized database where we keep all of this data, right?
Absolutely. What a great stepping stone for a PhD as well, developing that network. So for our early career scientists, great example there. This next question, I think, is a really great question. Alexia Dubuc asks, how were the different thresholds determined for hypoxia severity?
Yeah, that's a great question. Well, the 2 milligram per liter, 61 micromole per kilogram one is kind of the universal one that's been used in the literature since the 80s. And the upper limit that we chose was based on that meta-analysis of temperate species data, where they found that the 5 milligram per liter kind of encompassed 90% of the sublethal effects for all of the species that they studied. So we kind of felt like that was a reasonable upper limit. But this is really just in the absence of relevant data for tropical species. Like we would have based it on more quantitative studies of tropical species, but unfortunately there just aren't enough of them. So this is based, of course, on temperate species, and we wanted to have, a level between the most severe and the weakest. And so we just spaced out the other two between those. But this is not meant to say that these are the thresholds that, are important. These were kind of just placeholders, and hopefully the scientific community can help fill in the gaps and tell us, which of these thresholds are relevant and in what circumstances and perhaps for which reefs.
Absolutely. Sarah Stewart asks, would you expect the lethal hypoxic threshold to become either more or less constrained depending on the life stage of the organism, larval versus juvenile versus adult, for example?
Oh, that's a great question. I know there are at least a couple studies using like larval corals and looking at their hypoxia tolerance. I don't remember the levels off the top of my head, but I'm happy to look into that if you want to contact me after. But yeah, I don't know. I imagine they may be more sensitive as larvae, but I also don't study larvae, so I don't want to say for sure.
Yeah, I know with my team, we sort of look at those bottlenecks with different environmental stressors, being sort of at the early life stages or, during reproduction. And so that's quite relevant to some of the work that we do. Speaking of which, Erin wonders, what is that underlying mechanism that corals utilize to make them more or less tolerant to hypoxia that might drive that community structure that you observed? And is there any evidence of genotypic resistance to hypoxia in corals?
That is also a great question and part of some ongoing research that I know other people are working on. And I'm working on another project looking at some genotypic responses. And, I don't think that it's been totally identified. I know Rachel Alderdice, who was working with in Sydney, was also working on some of this stuff. So her work may be more informative for answering this question. But yeah, I think there's a lot of factors that go into it. And of course, there's like the boundary layer aspects of corals and seawater. And yeah, I don't know. I don't think we figured it out totally, but I think there's some amazing people doing research on that right now too.
Good research leads to more research, hey? I guess sort of the last question that I have kind of rounds back out to having working groups around the world. Are there oxygen data available for the Red Sea and the Gulf of Aden? And so this might be something that we really want to talk about as a community. And I encourage all of you who have put questions questions into the question box to please reach out to Ariel. She's got all of her contact information here on the slide because we have a really great opportunity as a community here to develop some working groups and to try to start answering some of these questions that we have remaining, which there are a lot, about how coral reef ecosystems are coping with oxygen issues. So I'm going to end the question and answer session for Ariel's talk now, and please absolutely join me in thanking Ariel for such a phenomenal talk. Really, really impressed as an ECR, Ariel, and I can't wait to see how things go for you over the next little while. So thank you so much for joining us.
Thanks for having me.
So I'll ask David to please share your screen. So David is our next speaker here in our webinar. Professor Suggett is also based here in Australia. Dave is also the president of the Australian Coral Reef Society, which is the oldest coral reef society in the world, celebrating our 100th birthday this year. Dave is also the team leader for the Future Reefs program and part of the climate change cluster at the University of Technology in Sydney. And so without further ado, Dave, please share with us your amazing research.
Thank you, Jodie, and welcome to everyone again. It's a real pleasure to be able to talk about all of the work we have been really undertaking, I guess, for the last 3 or 4 years now in the space of deoxygenation. I want to also say a huge thanks to Ariel as well. What a wonderful presentation and a great insight into some of the questions really that stimulated our research. And I think some of the questions that were posed in the chat, I think will hopefully give some insights over the next 20 minutes or so. And before I start, I do want to extend my deepest respect, acknowledgement and recognition to all traditional owners that we have been fortunate enough to work with and whose lands, sea country and land country, we've been working on for the past 3 or 4 years, and certainly to the elders, both past, present, and emerging. The recognition that we've had in this space has been incredible, and today I'm presenting from Darawal country. I also want to really acknowledge those that have driven this work, and especially two really exciting early career researchers, Dr. David Hughes and Rachel Alderdice, who have been part of a team coordinated by Michael Kuhn and Copenhagen, Chris Volster in Constance, and Matt Panisse, Emma Camp, JB Rayner, myself at UTS. And it has become a huge collective effort, as Ariel would have already alluded to, that we're growing as a community in this space. And many of our questions to really understand deoxygenation, as I said, were stimulated through the growing emergence of hypoxia events causing mass mortality on reefs, and really the pioneering work of Angelo Altieri and Maggie Johnson in the Bocas system in Panama. And this led us to really critically ask some questions, not only from the perspective of acute events where we have low oxygen waves appear on reefs and cause catastrophic mortality in much the same manner as we experience heat wave effects through temperature, but really also to consider the fact that we've got a bit of a menace in the room, and we know that reefs have already, or oceans have already lost oxygen content in the last 50 years. So what does that really mean for reefs in terms of much more subtle effects that are perhaps going on behind the scenes and what we call the silent squeeze? And it led us to really ask a series of questions as well that were really untapped amongst the community, and ones that really help frame our research priorities. And I've listed them here, and you can read them for yourself in your own time. But some of the critical questions we had was really to understand, is there capacity for corals to resist oxygen? If so, how do they do it? Is it heritable? And most critically, how is that informed by the oxygen history? And of course, Ariel's work is beautifully unpacking that for us in real time. And I'm really excited to see that paper come online soon, hopefully. And with that in mind, again, some of the issues that stimulated this questioning was we'd been fortunate enough to work in some really extreme systems that housed really quite elaborate reefs and reef formation, and one of which is demonstrated here. This is the Boraki semi-enclosed lagoon in New Caledonia. And what you can see on the right-hand side is some of the sort of physical metrics that we measure over time. And again, it has this characteristic diel variance in most parameters. Some of them are tidally driven, some of them are driven by, obviously, night-day. And the critical point here is, if you look at the dissolved oxygen lines, you can really see within the lagoon where we have thriving formations of organisms, branching corals that would typically die on a normal reef exposed to these sorts of conditions. The oxygen routinely goes down to 2 milligrams per liter, if not below. So the sort of classical hypoxia thresholds that we would consider. So we know that corals can withstand periodic exposure to low oxygen. And really the question for us was, how on earth do they do that? And consequently, what hardware do corals need to equip themselves on coral reefs to be resistant to deoxygenation into the future? This became sort of a little bit more complex once we started to think about corals themselves. They're extremely complex organisms. They are comprised of a heterotrophic host that is consuming oxygen all the time, but it's in association with a symbiont algae, which is of course releasing oxygen during the day. So it's actually subject to extremes in oxygen every single minute of the day. Some work that one of our PhD students, Nikki Delaney, has been performing is really thinking about how we translate what we measure externally into actually in the interior of corals once you have more complex branching systems. And this is just one example to show. You can see on the left-hand side here how oxygen variance is extremely amplified as you go deeper into a complex coral colony. Of course, not all corals form these complex colonies, but it gives you an example of just the extreme measures that corals are having to deal with on a day-by-day basis once you go beyond the external environment itself and into the boundary layers. That becomes more extreme itself. This is some classic work that Nikki is also taking forward that was initiated by Michael Kuhl really quite a long time ago now, but it demonstrated using microsensors that and you can see this on the right-hand side figure, the left-hand plot. This is the microsensor going deeper into the tissue. And as you go past the diffusive boundary layer, the open symbols demonstrate during the day it's hyperoxygenated as you go towards the tissue because of photosynthesis. But at night, that actually goes into anoxia. So corals are actually having to deal with zero oxygen at night. So of course, how on earth are they doing that? What this actually also tells us is that corals are really living already on the edge in terms of their oxygen exposure. They're really dealing with quite catastrophic exposure to highs and lows of oxygen during the day. So what we argue is that subtle variations in that exposure can have a very, very big effect indeed. And again, something that we are really not considering at the moment through this idea of the silent squeeze. And Rachel Alderdice really undertook some pioneering work during her PhD with us, where we took a very simple experimental framework. And the idea was, we know during the day that oxygen builds up in coral tissues. During the night, that's gradually consumed until it reaches anoxia. We call this the nighttime oxygen buffer. And the question we had was, what happens if we slightly take that away because of a deoxygenation event? And so what you can see on the left-hand side here is two different coral species, two Acropora taxa that dominate many of our reef systems. They've obviously been impacted hugely by heat waves, certainly on the Great Barrier Reef. They're starting to recover now. But the point is, it can be extremely successful to stress. And we expose them to both their routine declines in their nighttime oxygen buffer, but also with an eroded nighttime oxygen buffer. And then we sampled at several time points to capture how the corals were responding. And what was really interesting, and we weren't expecting this at all, is that one of our species remained phenotypically tolerant to this stress and did not bleach, whereas one of the corals, the coral on the right-hand side here, bleached during this process. So again, it told us that different taxa are inherently front-loaded to tolerate this kind of a stress, some more than others. And what Rachel managed to demonstrate was that, first of all, corals retain a metabolic network for hypoxia tolerance that's the same as we would find in as far as mammals. But similarly, what are the key mechanisms or the metabolic molecular repertoire that's being triggered during this? And you can see the diagram on the right-hand side. And just to synthesize this, there's a lot's taken on the right-hand side here, is that there's a lot of crosstalk between the same metabolic repertoire that drives deoxygenation stress bleaching as that we already know drives thermal bleaching. And so not surprisingly, we suspect that deoxygenation exposure is triggering some of the same cascades that we already know are well described for heat stress. And of course, that starts to ring alarm bells when we know that heat stress itself inherently lowers oxygen in our waters. So we have a reasonably good description now of the metabolic basis for deoxygenation tolerance or susceptibility in corals, at least at a molecular level, and certainly what genes are required to withstand that and potentially be selected for. We took that a stage further during the series of experiments, and we actually looked at larvae as well. And larvae that don't contain symbionts, some do, some don't. It depends on the taxa and the nature with which they reproduce on reefs. What we were able to look at here is that larvae that don't inherently have zooxanthellae within them when they are maturing within the water, they have no nighttime oxygen buffer. So they are inherently susceptible to extremes at night of low oxygen, especially once they settle down towards the reef benthic interface, where we know oxygen levels are incredibly low, if not anoxic at night. We conducted a similar experiment to the previous exposure with adults, and using actually the same taxa of one of the species that we knew was susceptible. And again, I'm going to keep this fairly short, but in the bottom hand little diagram here, it's safe to say that the larvae and the animal host, if you like, exhibited exactly the same metabolic response as we saw in the host. So we know again that larvae as a host are tolerant or susceptible to oxygen stress through their front-loaded metabolic repertoire. But what was most alarming in this bit of work was that simultaneously, some of the same triggers suggested that developmental factors were being impacted. So what we hypothesized in this paper, and something that still needs to be tested, is that developmental rates would also be a key outcome of deoxygenation in these systems. So again, deoxygenation has these subtle effects that really are not yet well captured and well caught, but in this case, certainly cascade across life history stages of reef-forming corals. And I'll touch back to that towards the end. So all of this tells us there's very dynamic oxygen on reefs. Corals can be equipped and armed to deal with them. We now know that corals must safeguard from deoxygenation. They've had to do this for millennia. And the question again is, how do they do that? Yes, we now know that there are molecular cues at play that are really critical. But there's also a lot of other emergent physiologies that we need to account for that actually dictate how this manifests, whether we see corals survive or thrive or demise. And as we've been discussing really throughout this webinar, is that hypoxia thresholds for corals are really not well captured at all. The dynamic range that exists within really complex, diverse communities on reefs is really not well understood. So one thing that Dave Hughes worked at for a couple of years was really adopt hypoxia response protocols that have been well established in other organisms and apply that to a range of corals. And the premise is pretty simple. It's basically, as you enclose an organism in a chamber and let it deoxygenate, how well can the organism retain its respiration rate above the rate of oxygen decline? We call that the extent of oxyregulation. And what I've plotted here from Dave's paper on the left-hand side is a demonstration of that regulation value. And this is really critical because until this point in time, most studies had assumed corals were what we call oxyconformers. So you lose oxygen, corals simply downregulate their metabolism. We didn't see this at all. What we found is that all corals oxyregulate, and they do this to different extents. And that actually this extent of regulation, which is on the y-axis, correlated really well with the extent of the hypoxia thresholds. And you extrapolate all of these parameters through the modeling process of the hypoxia response curve. What's really interesting on this figure on the left-hand side is you can see there's a P damicornis in the bottom left-hand point, and there's a P acuta in the top right-hand point. Those two taxa were taken from different environments, but they're exactly the same genus and growth morph. So it tells us that there's likely strong both taxonomic and environmental selection going on for how this regulation can be achieved. When we start putting this new data into the grand scheme of how other organisms can oxy-regulate and tolerate hypoxia, we can actually show that of certainly the taxa and conditions we've tested this for, corals do as well as many other organisms. Now, that doesn't mean it's good news for corals and for coral reefs. Just as a quick aside, one way that we demonstrated that they can do this during these experiments is that they can expose their polyps, so they can hyperinflate and increase their surface area. There's also processes such as cilia beating that might be upregulated, and we're exploring those processes currently about what drives that capacity to oxyregulate. So this brings us to the point that we again argue corals are really close to some serious thresholds at the moment. They're really operating on the edge of oxygen exposure. So it only takes a little bit of deoxygenation to really run them into a sublethal operating system. And this is really what Ariel was talking about with the hypoxia modeling. You know, as we start to see climate warming and change the metabolism of reefs, we really are pushing corals below that point of comfort, and they really are on a fine line. And this leads us to some really interesting new concepts that we've been thinking about in terms of sublethal exposure, because it really is the sublethal responses that are the silent drivers at the moment. Sure, acute events can be really, really well observed and well captured, but it's the sublethal and more inconspicuous responses that are of concern to us. And this little schematic conceptualized by Dave and myself and others in the team really try and capture that. And the idea is quite simple. Corals have a finite amount of resources for growth, reproduction, stress tolerance. In other words, their fitness characteristics and how they thrive and survive. And under strong oxyregulation, there's lots and lots of energy available to be able to retain their metabolism and fuel that into growth, reproduction, stress tolerance. As you start getting into weak oxyregulation, oxyconformity, you tend to be in a position where you simply downregulate your metabolism. And of course, that can impact on the energy you've got available to compete as a benthic organism. Things become much more severe once you deoxygenate. That's in the lower panel. And the idea here is that, of course, oxyregulation starts running at a much higher energetic cost and taking away from growth, reproduction, stress tolerance. So yes, you might be tolerant to deoxygenation, but you grow less well, perhaps, or you reproduce less well, and certainly you'd probably be less stress tolerant to acute events. And similarly, with our oxyconformity, we simply see more downregulation again. And whilst we may see In this case, similarly, less growth and reproduction capacity. It comes also at a cost of declined metabolism. So in this case as well, there are all sort of biogeochemical costs associated with these particular processes and how they manifest in the community. So we really are at a critical point to resolve these issues, because they really will ripple into how fit corals can be moving forward as benthic competitors in reef communities. Just as a very quick aside, and it's really a shout out to one of our other postdocs, Caitlin Lawson, who's been working on some work with Dave Hughes. And she's been demonstrating that, again, when you run these sorts of sublethal processes and you really force the organisms to make choices with the energy they have available, you see all sorts of unexpected metabolic outcomes. In this case, Caitlin's been looking at biogenic gas emissions from corals. And we really see that when you deoxygenate but sublethally, we see no fitness costs per se with the corals. They survive perfectly well. But what they emit and how that operates as a signal or a biogenic gas within the community changes. So we have all these hidden signals that we're currently not capturing through this process. And that's just one example. So what this has really led to is that we are really thinking about refining our research priorities now, since we first proposed them a few years ago. And where we're at now is thinking about these sublethal effects. As I said, yes, acute events are incredibly important, but it's really the sublethal, long-term, more chronic, ongoing stresses that are of immediate concern to try and resolve. This really tells us what reefs will look like in the future through deoxygenation. And I've repeated some of the same points from version 1 research priorities here, simply to say that we still haven't resolved many of these at all well. We have literally scratched the surface, as we would have seen in the two presentations today. But really where we're at now is that identifying hypoxia thresholds for coral competitors and predators is probably as important as for the corals themselves. So for example, if algae are not impacted by deoxygenation, but corals are in a sublethal sense, well, of course, that makes them, algae, for example, much better competitors into the future. Similarly, coral invertebrates and coral predators, they may similarly be impacted by deoxygenation in much the way as deoxygenation. So perhaps we won't see any shifts in the balance of power between corallifer activity and coral growth itself. And this really brings us to thinking about trophic cascades and the ecological modeling that we really need to do. This also cascades through to thinking about fish interactions. So fish are really important for sitting inside corals at night, branching corals and acting as to oxygenate within branching corals. But of course, if fish metabolism is impacted, how does that impact their capacity to oxygenate corals at night? So there's all these sorts of trophic web responses that we do not have a handle on, that we simply need to resolve. And we're starting to gain very good ecological models, but how we can parameterize those through deoxygenation effects is well beyond our grasp currently. And then coming back to the point with cascading effects across life histories, we've touched on reproduction a few times in the last 20 minutes, really understanding how larval effects cascade through to adult effects, and as well as heritability is perhaps one of our key ongoing concerns, something that we're starting to tackle reasonably well now with thermal stress. But again, deoxygenation stress is currently well flying under the radar. So thank you very much for taking the time to listen today, and hopefully that helps unpack a little bit more of how we've been trying to understand some of these uncertainties and really resolve the landscape of where we need to go next. Please feel free to contact me if you have any questions, and hopefully we've got some time left still today to entertain a few more, and hopefully that answers some of the questions in the previous section as well. So thank you very much.
Wow, thank you so much, David, for that talk. It was almost on cue, flowing on so nicely from Ariel's talk, and even examining some of the questions that she was really hinting toward in her talk, and some of those that came up in the question and answer box as well during Ariel's talk. So really, really nice marriage between these two presentations. Thank you so much. So for those of you listening and watching, please place your questions for David in the question and answer box, and we'll try to address all of those. We've just had some amazing questions coming through. And one that I'm pretty excited about from Andreas is, I wonder if oxygen concentration or oxygen partial pressure is the relevant quantity for corals. The fish people, okay, like myself, tend to look at partial pressure that might come up under warming, and even though concentration goes down. And so we're much more thinking those partial pressures relative to the concentration. So what are your thoughts on that?
Yeah, that's going to take quite a bit of unpacking, I think. And it's a great example of where I think us as a coral community are well behind those in the fish communities, and certainly the physical oceanography, the biogeochemistry. We've certainly been taking quite a first order take on this, thinking about hypoxia thresholds and just oxygen availability. So how that manifests to partial pressure, I can't answer that question. I'll be perfectly honest. But what we are starting to see, of course, is that things such as flow and boundary layers, and really the sort of physical organization of corals, is so critical to how oxygen availability manifests from that in the water column to that actually at the tissue interface, where of course the metabolism is doing the heavy lifting and being impacted. So it is joining those dots between oxygen availability, partial pressure, and of course the physics that is probably the key uncertainty we have at the moment in terms of knitting together some of our more ecological and physiological viewpoints. As you can see, I'm padding a little bit because I don't know the answer.
Absolutely. I think that just really emphasizes how much we need to work together, because
Yeah.
those of us that are at that interface and, really trying to understand what coral reef ecosystems, for example, are coping with under these various stressors, not just deoxygenation, but with temperature stress as well, and the role of all the other organisms. One of the ideas that really resonated with me that you mentioned was the role of the fish in the coral at night, and how they might be playing a really big role in keeping the coral oxygenated at night when it would normally be going down at night as well. So, we've examined a lot of differences between nocturnal versus diurnal species of fishes and how they tolerate those varying conditions. And so, can you comment on that a little bit? Getting back to the fish,
Yeah, no, I think that's a great point, because this is sort of how some of this started in terms of us as communities networking, is that we've got such a disconnect, and yet there's so much knowledge out there. And I think the thing to impress is that what we're learning in terms of corals' tolerance is really the sort of cumulative oxygen dosage they're getting during the day. And it's that nighttime oxygen buffer is really critical as to whether they go anoxic, whether they aerobic versus anaerobic metabolic pathways after really do the lifting. So yeah, the capacity of fish and other organisms to upflow where we know flow is a lot lower. So when we think about the physical architecture of different corals, that's really important in terms of fish inhabiting corals to do that. There's, similarly, are corals also having a metabolic load within the corals themselves? So is there actually some sort of net gain, net loss as fish start to suffer from the effects of deoxygenation? Are we going to tip that balance from them being active swimmers and oxygenating corals to becoming active consumers, and perhaps exacerbating deoxygenation within or around corals? And again, it's those ecological connections that we have no knowledge of at all. And a shout out, I think, to the other invertebrate biologists there as well. Again, there's a lot of knowledge in terms of how deoxygenation impacts invertebrates, especially in temperate systems. And we know invertebrates are incredibly important to the fitness of corals in terms of corallivore activity or other sorts of nutrient cycling. So again, it's making those ecological connections beyond the benthic solitary organisms themselves, sedentary organisms themselves, is really important.
Absolutely. That interplay between all the different organisms. We might think that those that are tucked away in the reef at night that, could be helping the coral would also have to be quite hypoxia tolerant to those conditions. So, really kind of a perfect marriage between all these species working together. Emma has a question about the molecular responses to hypoxia. And she asks if you looked at anaerobic enzyme activity. Maybe you could comment on some of those mechanisms.
Yeah, so absolutely. The molecular repertoires definitely demonstrate that the anaerobic pathways are upregulated, as you would imagine. So there's several factors at play. There's the hypoxia-inducible factor, heat shock protein network that is critical in all of the hypoxia regulation pathways, whether you look at anything above sponges, basically. And simultaneously, there's things like the glycolysis pathways through anaerobic pathways that are important. So all of these are incredibly energetically consuming. So as we've said, that corals have a finite amount of energy at the end of the day, and we know other stresses erode energy storage. So whilst they might have capacity to use these networks, the question is whether they have the resources to use these networks. And that's probably the most critical interface we're at now. And this is coming to the point Ariel was mentioning, that understanding the interaction of stressors now, where deoxygenation fits within that is really critical. We know a lot about now how temperature and pH interact and potentially regulate some of the same pathways, but we suspect that deoxygenation may even be one of the master switches within that. If deoxygenation is really the trigger of all of this, then how the other stressors manifest is really governed by the oxygen they have. It's really as simple as that. So yeah, just to summarize, really, yeah, the anaerobic pathways and glycolysis is really critical.
Absolutely. Well, we will end the questions there, and thank you for that one, Emma. Anyone who has questions still, all of David's contact information is here on the screen. Lots of different ways to contact Dave and keep those questions going. We've had just such a really, really great couple talks today for this webinar, and I would very much like to thank Dave, and please join me in thanking him for this great seminar. So Dave, I'll ask you to stop sharing your screen.
Sure, sorry.
It's okay. And I will share my screen one last time. So again, I can't even emphasize how wonderful these two talks were, and how it was really great to get that coral reef topic to our community, especially with regard to deoxygenation and thinking about some of the issues that coral reef ecosystems, not just here in Australia with the Great Barrier Reef, but coral reef ecosystems worldwide have been suffering with climate change. We think of coral bleaching associated with thermal stress and ocean warming, but deoxygenation could really be that underlying issue that we need to be investigating quite a bit more. I think we've definitely emphasized that here today. So I want to thank all of you again for your active participation today. We really look forward to welcoming you again for our next webinar. And don't forget to head over to the GO2NE website to download the first issue of our Good News newsletter that highlights the work of our groups, our good and our gone groups, and our community with respect to ocean deoxygenation. Have a really great morning, day, or night, everyone, and looking forward to joining you all again next time. Thank you.